So, Bismillahirrahmanirrahim. Now we come to the topic of excitation of skeletal muscle. Now they are saying that what's a neuromuscular junction and how the impulses are transmitted, right? So, skeletal muscle fibers, they are innervated by large myelinated nerve fibers, and they will originate in large motor neurons in the anterior horns of spinal cord. What do I mean by this? That look at this diagram. This is a skeletal muscle and it is innervated by a nerve which is myelinated, and its motor neurons will be coming from the anterior horns of the spinal cord, right? And they are going to come and they are going to innervate the muscle fiber at its midpoint, and the point where this nerve fiber will innervate the muscle, this is known as the neuromuscular junction, the junction between the nerve and the muscle. So each nerve ending makes a junction which is known as the neuromuscular junction. Now they are saying that action potential that is initiated in the muscle fiber by the nerve signal travels in both the direction towards the muscle fiber ends. What do I mean by this? Let me tell. With the exception of about 2% of the muscle fiber, there is only one such junction per muscle fiber. So now what will happen? An action potential will come from the nerve to the muscle, and what will it do? It will innervate at the midpoint, and the action potential will travel in this direction towards its one end and in this direction towards its other end. So it will move in both the direction, and this is true for all except for 2%. Now we come towards the physiological anatomy of neuromuscular junction. Now we see that a large myelinated nerve fiber is coming to a skeletal muscle fiber and it is innervating it at its midpoint, as we saw. Now the nerve fiber forms a complex of branching nerve terminals and they will invaginate the surface of the muscle fiber. The entire structure is called a motor end plate. Let me show you. So the junction between the nerve and the muscle where it is taking place, it is known as the neuromuscular junction, and this entire structure, it is known as a muscular end plate, and this end motor end plate, and this end plate is the longitudinal section, and this one is the surface view. How you can see how it is happening. These are the muscles and this is the nerve fiber, right? So this is how the neuromuscular or motor end plate is looking in two views. So now what will happen? A junction between single axon terminal and the muscle fiber membrane and the invaginated membrane is called synaptic gutter or synaptic trough. Let me show you. So you can see over here, this is an axon and this is the synaptic gutter or synaptic trough, and this region of the membrane in which it is invaginating, the axon is invaginating, it is forming a gutter or a trough. Okay, and these, in the axon terminal there are certain synaptic vesicles, and in these a region, that is the postsynaptic membrane, we have a lot of mitochondria. I will tell you why the mitochondria are important. Now these clefts that are produced in the gutter, they are known as subneural clefts. Sub means below, neural means neurons. So below the neurons there are these clefts. Coming back to the reading, so invaginated membrane, the synaptic gutter or synaptic trough, and space between the terminal fiber and the membrane is known as synaptic space or synaptic cleft. Let me show you. So this thing which is invaginating, it is forming a gutter or trough, and the space between the axon and this trough, this space is known as the synaptic cleft or the synaptic space. Also, this space between this axon terminal and the synaptic gutter, this is 20 to 30 nanometers wide. Then we come to another thing. They are saying that at the bottom of the gutter there are numerous small folds. They are known as subneural clefts below the neuron clefts. Okay, these serve to increase the surface area. Okay, we know that there is a similar structure which is present in our intestines, and that is known as villi. So the area in which there are a lot of invaginations and there are a lot of clefts, they actually serve to increase the surface area so that the enzymes or other things can work efficiently. Now in the axon terminal, many mitochondria are present, and these mitochondria will produce ATP, and this is the energy source which is used in the synthesis of neurotransmitter acetylcholine. And acetylcholine is, as we know, it's a excitatory neurotransmitter. What it will do, it will excite the postsynaptic membrane. And now what will happen? The acetylcholine will be packed in the form of small synaptic vesicles, and these vesicles about 300,000 which are present in the axon terminal of a single motor end plate, right? In the synaptic space, there are large quantities of the enzyme, and this enzyme is known as acetylcholinesterase. What will happen now? That this enzyme will destroy the acetylcholine after a few milliseconds, because if acetylcholine, which is an excitatory neurotransmitter, it will persist in the space, what will happen? It will lead to convulsions, or it will lead to a lot of random movements that are like this, movements like this taking place in our body. So what happens? So this was the axon terminal. It is having a large number of mitochondria. These mitochondria will produce ATP. These ATP will be used in the generation of acetylcholine. The acetylcholine is packed in these vesicles, synaptic vesicles, and now these synaptic vesicles, they will release their acetylcholine via exocytosis into the synaptic space, or the synaptic gutter, or synaptic cleft. They will release them into synaptic cleft. In the subneural cleft, now we will have certain acetylcholinesterase enzyme, and these enzyme, what will they do? They will degrade that acetylcholine, so a normal good or a smooth movement can take place, and that movement needs to end after some time. So now they're saying secretion of acetylcholine, how does it take place? So when a nerve impulse reaches neuromuscular junction, 125 synaptic vesicles containing acetylcholine, they are released. Now what will happen? Okay, before I tell you something else, let me show you this diagram. So coming to this diagram, you see these are synaptic vesicles. So this was the axon, this was the postsynaptic membrane, this is the synaptic gutter, this is the subneural cleft, and this area is known as synaptic cleft or synaptic space, right? So these synaptic vesicles, 125 of them are released on a coming of single nerve impulse. So single nerve impulse causes 125 synaptic vesicles to do exocytosis, release these acetylcholine neurotransmitter. Now what is this blue thing and what is this yellow thing? This blue thing is a dense bar, and on the sides of the dense bar we have calcium channels. They are very significant, let me tell you. Coming to a quick read. So on the inside of the neural membrane, there are dense bars, the blue ones that I showed you. And to each side of the dense bar, there are protein particles that penetrate the neural membranes. And these protein particles are what? They are actually calcium channels, voltage gated calcium channels. When an action potential spreads over the terminal, these channels open. Now what will happen? The action potential will cause the calcium channels to open. The calcium channels will diffuse into the synaptic space and to the interior of the nerve terminal. What will happen? Calcium calmodulin independent protein kinase will be activated. Let me show you. So what will happen now? The action potential will come. It will cause the calcium channels to be activated. The calcium channels will pull the calcium from this synaptic space to the nerve fiber. And what will happen? It will actually activate something. That something is calcium mediated protein kinase. What will this protein kinase do? This calcium calmodulin dependent protein kinase, which will phosphorylate the synapsin. Synapsin is actually protein that is anchoring the synaptic acetylcholine vesicles. So when the calcium calmodulin dependent protein kinase would phosphorylate those synapsin, so it will break the connection and it will free the acetylcholine vesicle. The free vesicles will now, what will they do? They will move to the active zone of the presynaptic neural membrane that is close to the dense bars. Now what will happen? These vesicles dock at the release sites and they release their substances by exocytosis. So let me tell you what happens. So an action potential comes, this action potential, what will it do? It will cause the entry of the calcium ions at this area. So calcium ions will now enter inside. Now these synaptic vesicles, they are attached by a protein synapsin. So these calcium channels will allow the calcium ion to move in. They will activate the calcium calmodulin protein dependent protein kinase. What will this do? It will break the connection, the synapsin, by phosphorylating it. What will happen now? This vesicle would be now free, and it will now come to this area adjacent to the dense bar, and it will do exocytosis and release acetylcholine into the synaptic gutter. This is how and what actually happens over here. Now, although some of the aforementioned details are speculative, it is known that effective stimulus for causing acetylcholine release is entry of the calcium ion, which is caused by the coming of action potential. Now, the acetylcholine from the vesicles is then emptied through the neural membrane adjacent to the area of the dense bar into the synaptic space. Coming to this diagram again, so you see a lot of synaptic vesicles that were attached with the protein synapsin. Now they have been degraded by the entry of the calcium ions. So in the subneural cleft, we see a lot of substances. What are these? These are acetylcholine receptors, and below these are voltage gated sodium channels. What will happen now? So acetylcholine will bind on these acetylcholine receptors, and it will cause the entry of the sodium ions. These sodium ions will decrease the negativity inside the membrane or increase the positivity inside the membrane. So what is now going to happen? Sodium will come in. It will become positive. This positivity will activate these voltage activated sodium channels. Now these voltage activated sodium channels will be activated. They will allow more sodium to come in, and when more sodium will come in, the membrane, postsynaptic membrane, will be depolarized. And what will happen? This now action potential, the membrane will be depolarized, and an action potential will travel in both directions, in this one direction and in this one direction as well. Let us give us a read. That acetylcholine opens ions on postsynaptic membrane. How? So, small acetylcholine receptors and voltage-gated sodium channels, acetylcholine receptors, voltage-gated sodium channels are present in subneural cleft. The acetylcholine-gated channels are located in the mouths of the subneural cleft. These acetylcholine are in the mouth of the subneural cleft, just below the dense bars, where acetylcholine is emptied into the synaptic space or the gutter. These voltage-gated sodium channel also line the subneural clefts. Simple as that. Each acetylcholine receptor, what is it? It's a protein complex. Now we see this calcium channel was also a protein channel. This acetylcholine receptor, they are also protein complexes. The receptor has a total molecular weight of 275,000. Fetal acetylcholine receptor complex is composed of 5 subunits: 2 alpha protein, beta, delta and gamma protein. It had alpha, beta, gamma and delta proteins. That was 5 subunits, fetal acetylcholine. But in the adult one, an epsilon 1 is added instead of gamma. So the adult one will have 2 alpha, 1 beta, 1 delta and 1 epsilon, and gamma will not be present. It will be replaced by epsilon. These protein molecules penetrate all the way through the membrane. We are talking about this acetylcholine receptor right now, right? So they are lying by the side of the membrane. The channel remain constricted in this normal case until 2 acetylcholine molecules attach respectively into 2 alpha subunit proteins, and they will cause a conformational change that opens the channel. So now we will consider this as the acetylcholine receptor. This was 275,000 molecular weight substance, and it has 2 alpha, 1 beta, 1 delta and 1 epsilon in the adult one. Epsilon in the adult one. So now what will happen? The acetylcholine binding sites, these are actually 2 alpha subunits, and on these alpha subunits, what will happen? 2 acetylcholine molecules will bind. Now when these 2 molecules will bind, a conformational change will occur. This will open, the channel will open, and it will allow the entry of the sodium ions. So we will have 2 sodium ions entering this, and when the channel will open, it will allow a diameter of 0.65 nanometers to be open. This is good for large enough for sodium ions, potassium ions, calcium ions to move easily. All these sodium, potassium, calcium, they will cause membrane depolarization. Now, patch clamp studies have shown that one of these channels, when opened by acetylcholine, can transmit 15,000 to 30,000 sodium ions in 1 millisecond. This channel we read about, that was 275,000 molecular weight channel. It can allow 15,000 to 30,000 sodium ions in 1 millisecond. How big of a depolarization it causes. So, conversely, negative ions such as chloride, negative ions cannot pass through this channel, but positive ions like sodium, like potassium, like calcium, they can move, and large amounts of them can move in 1 millisecond. So they're saying, why do the chloride ions cannot move? They cannot move because of this large negative charge which is present on the inside or on the mouth of these acetylcholine receptors. Now they're saying, in practice, more sodium ions flow through the acetylcholine gated channels than any other ion for two reasons. Now they're saying that these ions, these acetylcholine channels, they are permeable to sodium, they are permeable to calcium, they are permeable to potassium. But why do sodium ions are more flowing than any other ions? Firstly, because there are only two positive ions present in large concentration on the inside. As we know, there are concentration gradients, and as we know that cell is a bag of potassium. So the movement of potassium against its concentration gradient, like from its low concentration to its high concentration, is easy. But the opposite is not easy. So potassium cannot move inside the cell very easily. So what they're trying to say, that sodium ions are more in extracellular fluid and potassium ions are more in the intracellular fluid, as cell is a bag of potassium. Second thing is, the negative potential on the inside of the muscle membrane, that is minus 80 to minus 90, pulls positively charged sodium ions that are present on the exterior to the interior, right? So main thing is this minus 80 to minus 90 millivolt charge, which pulls the ion present on the exterior to the interior. Also, it will not allow the interior ions to move outside. This is how it works. So this entry of the sodium ions, what will it do? It will create a local positive charge inside the membrane, and this local positive charge, it is known as the end plate potential. I told you that the entry of the positive sodium ions will cause the membrane or the cell which was actually negative to become positive. This positivity is known as the end plate potential. This end plate potential, it causes sufficient depolarization to what now will happen. The acetylcholine receptors, these acetylcholine receptors, which allowed the sodium ions to enter, because of their negative charges inside the membrane and because of their concentration gradient, they will now cause positivity or end plate potential, and this end plate potential will now activate these voltage activated sodium channels. Now when these voltage activated sodium channels will open, muscle membrane will contract. Now I told you there was something else over there. There has to be something to degrade this acetylcholine, and this is the acetylcholinesterase enzyme. The acetylcholine, once released into the synaptic space, it is now broken down by this enzyme. Where is this enzyme located? It is mainly attached to spongy layer of fine connective tissue that fills the synaptic space between presynaptic nerve terminal and postsynaptic nerve terminal. So what they are saying, that it is attached to connective tissue. So consider this area having some sort of connective tissue, and on this connective tissue we will have a acetylcholinesterase attached, and this acetylcholinesterase will now begin the degradation of these acetylcholine. So a small amount of acetylcholine diffuses out of synaptic space and then is no longer available to act on the muscle fiber. The short time that acetylcholine remains in the synaptic space, the few milliseconds, it causes such a large influx of the sodium ions in this time. It excites the muscle fiber. The rapid removal of the acetylcholine, it prevents continued re-excitation. I told you this movement, if there is no cholinesterase, we will have our hand shaking, our movements continued, and the movement will not cease. But acetylcholinesterase prevents the continuous muscle re-excitation and helps to shut down this whole mechanism until a new action potential comes. Now coming towards end plate potential. End plate potential, I told you, the potential that is produced due to the entry of the sodium ions from the acetylcholine receptors. The sudden insertion of sodium ions into muscle fiber when acetylcholine gated channels open causes electric potential inside the local area to decrease. So it will decrease from minus 80 to minus 90 to 50 to 75. It will go from negative to positive side. Let me show you this diagram, how good it is. So this diagram tells you that normally it is from minus 80 to minus 90, but now since the entry of the sodium ions takes place and it reaches its threshold, and action potential is elicited, and the potential goes to 50 to 60 positive side, from minus 90 negative side to 50 to 60 positive side. So this was the end plate potential. The end plate potentials A and C are too weak to elicit an action potential. Oh, they are talking about that diagram I will show you. But the B one is much stronger and causes sodium channels to open. So this A and this C, they are very, very weak. But this B, it's a very strong one and it reaches the threshold level and it causes more positivity, more entry of the sodium ion. This area is the acetylcholine gated receptor entry, and this area, this is when sodium ions are activated, sodium channels are activated, and this causes the action potential or depolarization to take place. So this region, a peak going upward, is depolarization or muscle excitation. Now they're saying the weakness of end plate potential at point A was caused by poisoning, and this poisoning was by curariform drugs, a drug that blocks the gating action of acetylcholine on acetylcholine channels. So a drug which blocks the attachment of acetylcholine to acetylcholine receptor channels, it is known as a curariform drug. Now they're saying the point C that we saw, it is by botulinum toxin. It's a bacteria, and this bacteria decreases quantity of the acetylcholine released by nerve terminal. Let me show you what they're saying. So this A was curariform drug. Curariform drug will not allow the acetylcholine to bind to this acetylcholine binding site. And second one is botulinum toxin, that will actually limit the amount of acetylcholine being released into the synaptic cleft. But this one is normal. Now every part of the body, it has some safety factor. Ordinarily each impulse that arrives at the neuromuscular junction, it causes 3 times much more end plate potential as that is to be released. So this 3 times more action potential is a safety factor. Stimulation of nerve fiber at rates greater than 100 times per second or several minutes diminishes this. So what they are saying that actually one action potential causes 125 vesicles to be released, and this 125 vesicles, they are 3 times more than the amount that is actually need to excite the postsynaptic membrane. Understood? And now they are saying that if the re-excitation is 100 times per second, what will happen? The muscle, the postsynaptic membrane, it will be fatigued. It will no longer be able to process all those re-excitations or do the excitation again and again because it's very large. This situation is called fatigue of the neuromuscular junction, and it is the same effect as fatigue of the synapses in the central nervous system. Under normal function, measurable fatigue, it occurs rarely because it's a large number of the impulses coming per second, and they are not humanly possible except for electric shocks in which they become 1,000 times per second. So muscle fatigue, the postsynaptic membrane fatigue occurs in this condition, and the safety factor for production is 125 vesicles, which is 3 times the end plate potential which is actually needed. Now we come to the acetylcholine formation. It's very easy. Now let's come to this side. So they are saying that small vesicles, which are about 40 nanometers, they are formed in Golgi apparatus of cell body of motor neuron. So what they are saying, so we have this as, consider this a motor neuron, and motor neuron has a cell body, right? This is the cell body of the motor neuron. I'm trying to make a neuron. So this is the cell body, and this was the nucleus, and the cell body has a large number of Golgi apparatus. So the Golgi apparatus will produce acetylcholine because it has to produce what? Proteins, right? So Golgi apparatus in the cell body of the motor neurons and spinal cords, they will produce small vesicles which are 40 nanometers in diameter. These small vesicles will move through the axon. So the Golgi apparatus cell body produced small vesicles which are 40 nanometers from its Golgi apparatus, and they will move through the axon to reach the presynaptic membrane. About 300,000 of these small vesicles, they will collect at the presynaptic membrane near the skeletal end plate. Now, where is acetylcholine produced? It is produced in cytosol of nerve fiber terminal, but it is immediately transported through the membrane into the vesicles. Now, each vesicle will contain 10,000 molecules of acetylcholine. So, what will happen? Told you, this was cell body. Cell body had Golgi apparatus producing vesicles. These vesicles will move through the axon to reach the motor end plate. So this bulb of the axon, and this was the skeletal muscle cell. It will reach over there, and in the cytosol, what is produced? The main thing that is produced in the cytosol is your acetylcholine. This acetylcholine, 10,000 of this acetylcholine, they are packed in a single synaptic vesicle. 10,000. Now, the same mechanism explained. Action potential will arrive. It will cause the calcium channels near the dense bars to open. The calcium channels, what will they do? As a result, calcium concentration inside the terminal will increase about 100 folds. The calcium calmodulin mediated protein kinase will be activated, synapsin will be phosphorylazed, and exocytosis will take place, and 125 vesicles will be released, and they will cause sodium entry. The sodium and they will attach to the acetylcholine receptors. The acetylcholine receptors will cause entry of 10,000 to 35,000 sodium ions. Now the acetylcholine, it is split by acetylcholinesterase enzyme into acetate and choline ions. So what will happen? The number of vesicles available in the nerve ending, what will they do? They are usually sufficient to allow transmission of only few thousand nerve to muscle impulses. Therefore, for continued function, we need new vesicles, and these new vesicles must be reformed rapidly. Within few seconds, each action potential is over. Coated pit appear in the terminal nerve fiber. What will these coated pits do? They will do contractile proteins, and they have the contractile protein known as cladrin, which is attached to the membrane, and within 20 seconds, the protein contracts, the pits break, and form new vesicles again. Within another few seconds, acetylcholine is transported to the interior of these vesicles, and then they are ready to be released. Let me show you this whole cycle in a small flow chart. This diagram of mine. So what will happen? An action potential will come. Let's skip that part. Let's go to how acetylcholine is produced. So this is a Golgi apparatus. It will produce vesicles 40 nanometers in diameter. From the cell body, they will move through the axon to the axon terminal. In the axon terminal, in the cytosol, there will be acetylcholine produced. This acetylcholine will move to the vesicles. 10,000 is filled in one vesicle. Now, when calcium ions come in the dense bars, they will produce all the things that I explained priorly. You can rewind the video and see. Now, this acetylcholine is released in this area. This acetylcholine then causes the activation thing that I explained earlier. Then what will happen? Acetylcholinesterase will break it into acetyl and choline. This acetate and choline will now, what will they do? They will be repacked by cladrin proteins. The cladrin proteins will again form the vesicles, and they will move to the interior. This is done to actually conserve the amount of the vesicles, and hence have a smooth flow, and so to prevent any fatigue of the neuromuscular junction. and this cladrine protein is occurring within 20 seconds. So now we have drugs that are similar in function to acetylcholine. We know acetylcholine is a what type of drug? It is a activatory or excitatory neurotransmitter. So other compounds like methacholine, carbachol, nicotine, they have similar effect to acetylcholine. So what is acetylcholine effect? Depolarization of the postsynaptic membrane. So these drugs work by causing localized areas of depolarization of the membrane of the motor end plate. So then every time muscle recovers from a previous contraction, the depolarized area by leaking of the ions initiate new action potential, and since these cause repetitive depolarization, they cause muscular spasm to take place. Now there are certain drugs which inactivate this enzyme that breaks down acetylcholine into acetate and choline. These drugs are neostigmine, physostigmine and diisopropyl fluorophosphate. What will they do? If acetylcholine is not broken down, repeated again and again excitations or re-excitations will take place, and they will produce same effect, that is depolarization areas and muscular spasm. And if the spasm goes to your which apparatus, that is your respiratory apparatus, it will lead to laryngeal spasm and person might die. Neostigmine, physostigmine combined with acetylcholinesterase and it will inactivate it up to several hours, but they can be removed from the body. They are removed after some time. But there is one thing, that is diisopropyl fluorophosphate, which is also known as nerve gas poison. It inactivates acetylcholine for weeks, and when there is for weeks the muscular spasm, it goes to laryngeal spasm and the person might die. Now I told you that was the A thing on the graph. It was done by curariform drugs. And what did the curariform drugs do? They did not allow acetylcholine to be attached to its acetylcholine receptors. So they will prevent the passage of the impulses from nerve ending into the muscle. So there is a example, D-tubocurarine. It blocks the action of acetylcholine on the muscle fiber choline receptors. No attachment to receptors, preventing increase in permeability. So when acetylcholine is not attached, what will happen? No threshold is released, no threshold is reached, no action potential is produced, no depolarization takes place, and muscle is not activated or excited. Now coming to this autoimmune disorder, that is myasthenia gravis. It occurs in 1 in 20,000 people. Now what happens? Our own body tends to produce antibodies against the acetylcholine receptors that will present in the mouth of the subneural cleft. So when the receptors will be attacked by the antibodies, they will be destroyed. They will not be able to work. And what will happen? Postsynaptic neuromuscular junction would be destroyed. There will be no activation. Regardless of the cause, the end plate potential are too weak to initiate an action potential. And what will happen? The muscle fiber will not be depolarized. There will be no excitation. So what can we do to actually excite this thing? We can use drugs that had similar action to acetylcholine, or we can also use drugs that were going to block the acetylcholinesterase. These drugs were neostigmine, physostigmine and diisopropyl fluorophosphate. Within minutes, some of those areas that were affected, they began to function normally until the new dose of neostigmine is required. And we know this neostigmine, it occurs in our body for several hours. So new doses are required few hours later. So this is a very lethal disease, myasthenia gravis, and people need to take new doses of neostigmine or the drugs, because there is no therapy to prevent this. So they need to take this one, or else what will happen? They will die, because no action potential will be generated, no excitation will take place. They will lose their conscious control on their muscles. Now coming toward muscle action potential, we know the resting membrane potential was -80 to -90, right? The duration was 1 to 5 milliseconds in the skeletal muscles. and velocity is 3 to 5 meters per second. So now this minus 80 to minus 90, which is in the skeletal fibers, it is 10 to 20 millivolts more negative than neurons. So muscle is more negative than neuron. This 1 to 5 millisecond time is 5 times as long as in the neurons or large myelinated nerves. And this 3 to 5 meter per second, it is 1 by 13 the velocity that occurs in large myelinated nerve fibers. So skeletal muscles are slower than the nerve fibers. They have large duration of action potential as compared to the nerve fibers, but they are more negative as compared to the neurons. Now skeletal muscle fiber is so large that action potential spread along its surface. Maximum muscle contraction, however, requires current to penetrate deeply into the muscle fiber. Now, how it will penetrate deeply? There are some things. These are transverse T tubules that penetrate all the way through the muscle fiber. And what will they do? These T tubules will cause release of calcium ions inside the muscle fiber, and they will cause excitation contraction coupling. Let me show you this whole apparatus. So this is your myofibrils, and this is your T tubule, transverse tubule. And the transverse tubule, it is very thin and it will release the calcium ions from the exterior to the interior to each and every cell. So what will happen? It will cause excitation contraction coupling, and it will cause nerve impulse to be generated. We know that calcium ions entry causes acetylcholine release and all that stuff. So this is how the calcium ion is entering in the whole muscle. So now what will happen? The transverse tubule, the sarcoplasmic reticulum system. So where the T tubules originate from the cell membrane, they are open to exterior of the muscle fiber. I told you that they will push the calcium from the exterior to the interior. Therefore, they communicate with extracellular fluid surrounding the muscle fiber. In other words, T tubules are actually internal extensions of cell membrane. So what happens? The cell membrane, it has inward invaginations. These inward invaginations, they connect the extracellular fluid to the intracellular area and allows the calcium ion to move inside. So this what will happen over here? Therefore, when an action potential spreads over a muscle fiber, a potential change also spread along T tubule because they are cell membrane. So the electric current surrounding the T tubules then elicit muscle contraction. Sarcoplasmic reticulum is composed of two major parts: large timbers, the terminal cisternae, and long longitudinal tubules surrounding all the surface of contracting myofibrils. Let me show you this. So we have the sarcoplasmic reticulum, this yellow area, then the sarcotubules, and then the transverse tubules. They are explaining this only. So what release of calcium ion by sarcoplasmic reticulum? What will happen now? So now when an action potential reaches the transverse tubule, the voltage change is sensed by dihydropyridine receptors linked to the calcium release channels, also called ryanodine receptor channels. Activation of the dihydropyridine receptor or ryanodine receptor channel triggers the opening of calcium release channel in the cisterna. So what will happen? Let me tell you. So this diagram is your lifesaver. This nerve terminal. So when an action potential comes over here, it is sensed by the dihydropyridine or ryanodine receptor channels. These dihydropyridine receptors, they are linked to the calcium release channels in the sarcoplasmic reticulum. So the dihydropyridine receptors, when they sense an action potential, they will cause the opening of these calcium release channels. The calcium, it will move out from the sarcoplasmic reticulum into this area. Which area? This interstitial area. So these ryanodine receptor channels will open the cytoplasmic reticulum. So activation of dihydropyridine receptor triggers the opening of calcium release channels in the cisterna. So now what will happen? These channels remain open for a few milliseconds, releasing calcium into the sarcoplasm. Where was it released? Into the sarcoplasm. And now it will cause contraction. This is how excitation leads to contraction. Excitation by the Excitation by the action potential causes excitation of dihydropyridine receptor, which causes opening of calcium release channel, which causes release of calcium into the sarcoplasm. This calcium will cause operation of all the other things, and contraction will take place. So this is excitation contraction coupling. So now the calcium ions that have entered, they are supposed to be removed from this area. So calcium pump will remove the calcium ions from the area. And what sort of calcium pump is over there? That is the SERCA pump, that is smooth endoplasmic reticulum calcium ATPase pump. Come to this teeny weeny diagram. So what will happen? There is this SERCA pump, that is smooth endoplasmic reticulum calcium ATPase pump. It will use ATP, it will pump the calcium from the sarcoplasm back to the sarcoplasmic reticulum. This calcium will now attach to calciquestrin protein. So the SERCA pump can concentrate calcium ions about 10,000 folds inside the tubules. So what will happen? Now these calciums will attach to calciquestrin, and the calciquestrin ka single molecule can bind 40 calcium ions. So now they are discussing what is the difference of the calcium ion to the exterior and interior. They are saying that normal resting state 10⁻⁷ calcium ion is present in the cytosol. Therefore, troponin tropomyosin complex keep actin filament inhibited and maintains relaxed state. When full excitation takes place, the 10.7 molar, 10 raised by 7 molar, it increases to 2 into 10 by minus 4 molar, which is 500 folds increase. Now the troponin tropomyosin complex will be activated. Immediately thereafter, calcium pump, it will again decrease this back to 10 by minus 7 molars. So the total duration of the calcium pulse or increase of the calcium from 10 to the minus 7 to 2 into 10 to the minus 4, it lasts for about 1/20 of a second. During this calcium pulse, muscle contraction can occur. If the contraction is to continue without interruption, a series of calcium pulses may be initiated by continuous series of repetitive action potential. They are saying that to have repeated muscular contractions, we need to have repeated calcium pulses, and the calcium pulses is the increase or influx of the calcium ions. I always like a clinical. Now we have the clinical malignant hyperthermia. What is malignant? Malignant is something that tends to increase, and hyper is increased and thermia is temperature. So and a hypermetabolic crisis, it means increased metabolism. It is triggered by exposure to anesthetics like halothane, isoflurane, succinylcholine. There are 6 genetic mutations of the ryanodine receptor or the dihydropyridine receptor genes. Let me show you. This receptor, the DHP, the dihydropyridine or the ryanodine receptor, it undergoes certain mutations. And these mutations or these malignant changes, they are actually triggered by halothane, isoflurane and succinylcholine. So we will have hypermetabolism. These mutations cause unregulated passageway of the calcium ions to the sarcoplasmic reticulum. The dihydropyridine receptor, it is abnormal, it's abnormally working. There is calcium ion release from the sarcoplasmic reticulum to the sarcoplasm. So what will happen? There will be sustained muscular contraction. Again and again muscle will be contracting, generating large amounts of heat or increased metabolic rate or hypermetabolic syndrome. So what will happen? Hypermetabolism will take place due to faulty dihydropyridine receptors, which cause large amount of calcium influx. And because calcium is coming in, it's a positive ion, so it will cause acidosis, and it will decrease or deplete all the energy stored. All the ATP will be depleted. There will be acidosis, there will be increased body temperature, there will be hypermetabolism. So symptoms, they will include muscle rigidity, high fever, rapid heart rate. Additional complications, they include breakdown of the skeletal muscle starts to take place. Why? Because of high fever. High fever causes breakdown of proteins. So there will be rhabdomyolysis, that is breakdown of skeletal muscles. So now it can be treated by dantrolene, which actually antagonizes ryanodine receptors, and it actually is a blockade of the ryanodine receptor channels, so it will not allow the release of the calcium ions. So this malignant hyperthermia was triggered by isoflurane, succinylcholine, and halothane, but it can be treated with dantrolene, which will actually stop the ryanodine receptor channels from working. So there were two diseases in this chapter: malignant hyperthermia and myasthenia gravis. Please remember them, and all of your MSK1 physiology will be done for this chapter. Thank you very much, and I will meet you in some other chapter. If you like my style of teaching, please let me know in the comments below, and if you don't like it or you have some suggestions, please let me know as well. Thank you very much.